All right, we're going to get started. Thank you all for joining us today for today's session in the Innovators of Cognitive Neuroscience speaker series. Before beginning, we do want to acknowledge the indigenous lands from which we are joining today's talk. I am calling in today from New York City, or Mannahatta, which means hilly island, as it was originally called by the people from whom this land was taken, the Munsee Lenape and the Wappinger people. Through recognition of these past harms, we can step towards more equitable and humane futures. I encourage everyone to visit the link that I've just posted in the chat to learn more about the indigenous lands from which you are calling in from today. All right, hi everyone. My name is Victoria Shelkin, and on behalf of all of the students and postdocs on the organizing committee, I'm excited to welcome you all to today's Innovators in Cognitive Neuroscience seminar. The organizing committee together represents a consortium of colleges and universities, including Dartmouth, University of Pennsylvania, Yale, MIT, Princeton, Harvard, Columbia, and Gallaudet University, that are committed to highlighting advances in cognitive neuroscience research. We acknowledge that these schools are elite institutes of higher learning, so to make the science discussed here available and accessible to all, these seminars are open to anyone who's interested, and with consent of our speakers, we plan to make these talks publicly available on our website. So that brings me to today's talk. Today we are hearing from Dr. Sarah Yip. Sarah completed her Master's in Science in Psychodynamic Developmental Neuroscience at University College London in 2008, before completing her PhD in Psychiatry at the University of Oxford. Sarah is now an assistant professor in psychiatry at the Yale School of Medicine, and given Sarah's background, her current work falls nicely at the intersection of cognitive neuroscience and clinical psychiatry. Her work aims to develop brain behavior models of addiction and addiction treatment using neuroimaging-based predictive modeling and pharmacological studies in patients and in healthy controls. Today, Sarah will be sharing some of this work with us in her talk titled Towards Reliable Brain Behavior Models of Addiction Outcomes. This talk is being presented in support of the Integrated Refugee and Immigrant Services Organization. You can follow the link on the screen, and I will also post it in the chat if you would like to learn more about the work of IRIS and how you can support. Another note is that today's talk is being interpreted in American Sign Language. We have the interpreters spotlighted on Zoom, but if you would like to additionally see Sarah's video while she is presenting, you can choose the gallery mode option on Zoom. And last note before I hand things over to Sarah, everyone can please use the Q&A feature on Zoom to ask questions throughout Sarah's talk that we will address at the end of the presentation. And after this session is over at 1 o'clock, we are going to move over to a new Zoom room for an additional trainee Q&A session. This is a time for anyone to ask additional questions that weren't answered during the talk, or any questions about Sarah's career in science or otherwise. I'm posting the link to that Zoom room now in the chat, and I will also post it at the end of today's presentation. And with that, I will hand things over to Sarah so that we can get started.
Great, thank you so much for that introduction. All right, let me just navigate screen sharing here. Okay. Can you see my slides?
Yep, everything looks good.
Just my slides, no notes.
Yep.
Great. So thank you so much for the invitation to talk today. I'm excited to present some of my group's neuroimaging work that we've been doing over the past few years, really focusing on addiction treatment outcomes. I'm sure everyone here is aware of the current substance use epidemic in this country. I nonetheless think it's really important to mention at the start of the talk. So this color map is showing population level deaths per 100,000 individuals in the United States back in 2014. And as we know, these rates have continued to rise. So for example, annual opiate associated fatalities recently exceeded those caused by firearms and motor vehicles combined, as well as those caused by HIV at the height of the AIDS epidemic. And although it's been less publicized, there's also really been a concurrent rise in cocaine and stimulant associated fatalities. Therefore, improved strategies to combat the current substance use epidemic really are urgently needed, which is the motivation for my lab's neuroimaging research. I often get asked why we would even use neuroimaging to predict treatment outcomes in the first place. So after all, we're pretty fortunate in the addiction field. We have a large number of really excellent evidence-based treatments that are very effective for some individuals. However, we also have a lot of between patient heterogeneity, which unfortunately means that the overall efficacy of any given treatment tends to be highly variable across individuals. And so given that heterogeneity, it's perhaps not surprising that even gold standard treatments tend to have high rates of relapse, and that most individuals with a substance use disorder go through multiple prior failed treatment attempts. So in other words, when it comes to addiction treatment, one treatment really doesn't fit all. And then that problem is further exacerbated by the fact that what I like to call traditional clinical variables, so things like baseline severity, don't tend to be all that helpful for predicting treatment response or relapse rates, meaning that we have these unexplained sources of heterogeneity that are influencing treatment outcomes. And so within this context, a number of research groups, including my own, have turned to neuroimaging measures to try to identify brain-based predictors of treatment response. And over the past 10 to 15 years, there have been a number of proof of concept studies linking brain function and structure to treatment outcomes and addictions. So for example, work by our group and others indicates that individual differences in pre-treatment reward-related activations are prospectively linked to within treatment abstinence, that changes in functional neural circuitry may co-occur with reductions in substance use, and that individual differences in functional connectivity relate to both current cocaine use and future relapse. So collectively, these and other studies are really supporting this overarching hypothesis that individual differences in brain function are indeed linked to differences in treatment outcomes. However, this work also has some limitations. So in particular, most prior studies have relied on prospective associations and have used methods like correlation and regression that have a tendency to overfit the data, which can lead to inflated effect size estimates. And so really the problem with using the term prediction in this context of simple correlation or regression, is that true prediction really requires application of a model to novel data. As is the case when we use a machine learning approach, which seeks to reduce overfitting via creation of a predictive model in a data-driven manner using a training data set, and then application of that model to an independent test data set, which is then used for model validation. And that distinction is really important because whereas the aim of traditional statistics is generally to explain the variance between two variables, the aim of machine learning methods is to generate predictions in novel data. And so I would argue that this type of approach is really critical for the eventual translation of research findings into clinical settings, which is really a primary challenge of modern psychiatry. In addition, I would also argue that when used appropriately, these approaches can also be used for neurobiological discovery, which for my group is really important because, in my opinion, in addition to generating predictions and novel data, a primary goal of brain-based predictive models should really be to elucidate mechanism, because otherwise even highly predictive models often do little to advance our mechanistic understanding. So throughout the talk today, I'm really going to be trying to focus on maximizing neurobiological discovery within the context of predictive modeling. In order to maximize this kind of mechanistic discovery, we use data-driven whole brain methods. And the reason for that is pretty simple. It's that given that recovery from addiction involves complex interactions across clinical and biological domains, we think it's likely that achievement of abstinence also involves distributed processes from multiple different brain regions. So all of the data I'm going to be showing you today is using a whole brain connectivity-based approach in which we're focusing on patterns of functional connectivity across the entire brain. So the specific method I'm going to be focusing on throughout the talk is referred to as connectome-based predictive modeling. And unlike some other machine learning approaches, this is an entirely data-driven technique in that it doesn't require any sort of a priori specification of networks or regions of interest. And it's therefore both a predictive tool as well as a method of identifying novel networks. And so prior to the data that I'll be presenting today, this approach had been previously used to generate robust predictive models of trait behaviors such as IQ, but it had never before been applied to predict a future behavior or a clinical outcome. So when we use a connectivity-based approach, what we're doing is we're extracting time courses of activity from multiple regions that together encompass the entire brain, which are represented here by these cartoon green and red voxels. And what we do is we simply correlate these patterns of activity across the time course of the whole scan to get a single summary statistic that summarizes the temporal coherence between those two brain regions. And so we can do this for every single voxel in the brain to create a functional connectivity matrix. So this single matrix is now summarizing whole brain connectivity patterns for all of the voxels in the brain for a single person. And there's data to indicate that while these connectomes are relatively unique to the individual, patterns of connectivity within the connectome for a given individual may also vary as a function of task performance, or what we like to refer to as brain state. So consistent with this notion of brain state, prior work conducted by some of my colleagues here at Yale has demonstrated that actually the accuracy of predictive models generated from functional connectivity data is actually improved when the input is connectivity during task performance as opposed to during resting state. So here on the left, you have the percent variance of IQ that is explained by a predictive model built using different tasks from the Human Connectome Project. And then on the right, you can see the same thing for data from the Philadelphia neurodevelopmental cohort. And what I want to point out here is that in both cohorts, the predictive accuracy of models built using resting state data is relatively low. So in both cases, the model is only able to account for about 4% of the variance in IQ. The second thing I want to point out is that the predictive accuracy of models generated from different types of task data is also highly variable, with some tasks accounting for up to 12% of the variance in IQ and others accounting for much less. So together, these data are really suggesting that specific brain state manipulations may be preferable for predicting a given behavior. And that's an issue I'm going to return to throughout my talk. So that brings me to the data that I'll be presenting today, in which we're using this connectome-based approach to try to predict a clinical outcome. In this case, in the case of this first data, at least, we're trying to predict abstinence from cocaine during a 12-week treatment. So the study design for this is pretty simple. We recruited patients entering a 12-week treatment trial, and we scanned them at the start of treatment and then again following treatment. All of our participants were receiving methadone maintenance therapy for opiate use disorder, and now we're entering treatment for cocaine use disorder. So these are polysubstance using individuals. I'm not going to go into the details of the 12-week treatment trial today, but if you're interested, it was an RCT of behavioral therapy with and without treatment with a cholinesterase inhibitor. So consistent with the overall RCT, our neuroimaging sample for this was prominently male. They were mostly unemployed, and their primary route of cocaine administration was via smoking. The individuals in this trial also had a number of prior treatment attempts, including an average of 3 or more prior failed outpatient treatment attempts, as well as an average of 3 or more prior failed inpatient treatment attempts. And they also had significant legal problems. So really, this is a very treatment refractory population, but it's also a fairly typical one. So just to give you a schematic overview of the connectome-based approach that I introduced earlier, here we have a schematic overview. And so when we're doing this approach, practically what we're doing is we take individual connectomes from a training data set, represented by these gray and black and white boxes, and we relate them to a behavioral variable of interest. So in this case, we're relating our matrices to within treatment abstinence. And we do that just using simple correlation to identify positive and negative predictive connections, or edges. So positive edges are connections for which increased connectivity predicts abstinence, whereas negative connections are connections for which decreased connectivity predicts abstinence. And so this is really our feature selection phase. We then create individual participant summary scores via summing the edge weights identified in the feature selection phase, so that each participant now just has 2 values. One is a positive summary score and the other is a negative summary score. We then create our brain behavior model by entering those summary scores into predictive models, assuming linear relationships. So a simple y equals mx plus b. And then finally, we apply this model to our testing data set. So we take connectivity matrices from an independent data set. We extract connectivity values from the edges we identified in our training data set, sum them to create summary scores for our new participants, and enter those values into our brain behavior model to generate individual predictions of within treatment abstinence. And then finally, although it's not shown here, we of course evaluate the accuracy of our model by comparing predicted values with actual within treatment abstinence values. And we can do that a couple of different ways, which I'm not really going to go into today. The simplest is really rank correlation, which is what I'm going to present, use throughout the talk today. So for this study, we had a couple different types of functional data to choose from. So during pre-treatment scanning, everyone performed 2 tasks. One was a basic cognitive control task, and the other was a simple reward paradigm. And so given that we were interested in predicting cocaine use, we chose to use the reward task data because at that time, the data linking reward-related behaviors to cocaine use seemed more consistent than that linking cognitive control to cocaine use. And so using the data acquired during this reward task, we computed individual participant connectivity matrices, and we fed this into our connectome-based model, along with our measure of within treatment abstinence, which here is defined as the percentage of twice weekly urine specimens that were negative for cocaine during 12-week treatment. So this is a biologically verified dimensional measure of abstinence. So here we have our initial model results. Here on the y-axis, we have individual participant abstinence values as predicted by our brain behavior model. And on the x-axis, we have the actual abstinence values for each participant. So typically when we use correlation, we're trying to explain the variance between two variables, but here we're just using it to characterize predictive accuracy or the correspondence between actual and predicted values. And as you can see, our model has relatively good predictive accuracy with a Spearman's error of 0.42, which means that about 20% of the variance within treatment abstinence is accounted for by connectivity within our abstinence networks. And interestingly, that correspondence is greater than that observed if we just relate abstinence to another clinical variable, including baseline cocaine use. And really that's indicating that our brain behavior model has greater predictive accuracy than traditional clinical variables. So in addition to generating behavioral predictions, this approach, of course, also identifies networks, which I'll show you in a second. As a reminder, networks identified using a whole connectome-based approach are typically complex, and they can be composed of multiple adjacent and non-adjacent brain regions, which was certainly the case with our networks. So here on your left, you can see the positive network shown in red. As a reminder, the positive network includes connections for which increased connectivity is predicting abstinence. And then on the right, you can see the negative network, which corresponds to connections for which decreased connectivity predicts abstinence. And so while these networks are definitely complex and they're arguably pretty hard to interpret when presented in this fashion, I think it's really important to point out that actually the combined number of connections within the positive and negative network is only around 500, which is actually less than 2% of all possible connections using this parcellation scheme. So in other words, despite this visual complexity, these are actually really quite specific connections. And so there are a number of standard ways in which we can then start to summarize these connections to facilitate interpretation. So these circle plots are summarizing network connectivity based on the number of connections between macro scale brain regions, which are listed on the left. And then from top to bottom, the brain regions for each circle are represented in rough anatomical order, with lines coming from the red portions at the top corresponding to prefrontal cortical connections, and lines coming from the purple sections at the bottom corresponding to brainstem connections. And then from left to right, connectivity is thresholded at different levels based on the number of total connections per brain region. So you can see that if we use a liberal threshold, so for example, if we look at all brain regions with 5 or more connections shown on your left, our networks remain somewhat difficult to interpret. However, if we take a more conservative approach and we only focus on regions with 12 or more connections shown on the right, our networks begin to be a bit more interpretable. For example, now we can see that the positive abstinence network shown in red at the top is characterized by a right prefrontal node with connections to temporal, limbic, and left prefrontal cortical regions. And then we can also see that the negative network shown at the bottom is characterized by a temporal node with connections to limbic and cerebellar nodes. So another way that we can also begin to understand the anatomy of our abstinence networks is just via their spatial overlap with established canonical networks. So for example, via their overlap with things like the default mode and frontal parietal networks. And so when we do this, we can then characterize connectivity based on the number of connections between these established networks. So here we have matrices that are summarizing overlap with these canonical networks for the positive and negative abstinence networks separately. And for each matrix, the cells represent the total number of connections within and between each network, with the darker colors indicating a greater number of connections. And when we do this, we can see that the positive network is characterized by a large number of frontal parietal and medial frontal connections, as well as by a lot of salience and motor sensory connections, as indicated by these dark red boxes. And we can also see that the negative network includes a large number of salience, default mode, and medial frontal connections, as indicated by the dark blue boxes. And these differences become even more apparent if we directly compare the number of connections within each network. So when we do this, we can see that the positive network includes more frontal parietal to medial frontal connections, as well as more salience to subcortical to motor sensory connections. In contrast, the negative network is including more connections between the medial frontal network and salience and default mode connections. And so based on these differences, we've generated a theoretical network-based model of cocaine abstinence. So based on our data, we propose that cocaine abstinence is positively predicted by increased connectivity within and between frontal parietal and medial frontal networks. As a reminder, these are networks involved in coordination of attention and executive control. And so we think that these might contribute to within treatment abstinence via coordination of things like top-down processes that are necessary for treatment engagement. So things like acquisition of new skills or enhanced control over impulsive behaviors. Our data are also suggesting that cocaine abstinence is positively predicted by increased connectivity within salience, subcortical, and motor sensory regions. And these are really networks that are involved in things like coordination of salience encoding and reward. And so we think that these networks might support motivational processes relevant to treatment. So for example, things like willingness to change or shoring up of non-drug reward processing and attending to alternate rewards. Finally, our data is indicating that abstinence is further predicted by decreased connectivity between these two systems. And so based on prior connectivity work in cocaine use disorder, we actually think that this appropriate separation between these two systems might relate to greater behavioral flexibility or decreased compulsivity, as would be required for behavior change during treatment. So this model is really building on prior models of addiction that emphasize separation of frontal parietal and salience networks, but it's also incorporating medial frontal, motor sensory, and subcortical networks to really provide a theoretical framework for future research. So really our hope is that by proposing this framework and summarizing our data in this somewhat simplistic way, we can encourage others to test these hypotheses in their own data. We've also publicly shared all of the actual masks and code for these analyses to again, facilitate people testing these hypotheses in their own data. So before I move on, the last thing that I want to point out about this schematic is that if we look at the networks that are being highlighted, what's interesting is that even though when we create our connectome-based model, we're actually just using the time course of activity over the entire task. We're not modeling specific events of interest. But despite that, the networks that are emerging as predictive of abstinence seem to be somewhat intuitive within the context of the reward task that people were performing, because performance of this task really requires coordination of both attentional and cognitive control processes, as well as salience encoding and reward response behaviors. So this is just like a really early hint that the brain state might matter for prediction, which again is something I'm going to sort of return to throughout the talk. So we've also tested the ability of the network that we identified to predict abstinence following treatment. So for this analysis, we simply create summary scores by summing edge weights for the nodes identified in our original analysis. So we're taking our abstinence networks and using them as a mask to extract values from individuals who are scanned 3 months later, so at the end of their treatment. And then we enter those data into correlation analyses with their % days of abstinence during 6-month follow-up. And when we do this, we find that post-treatment network strengths are in fact significantly correlated with post-treatment abstinence, suggesting relative consistency of this relationship over time. Further consistent with that, when we just compare pre versus post-treatment network connectivity, we don't see any changes over time, sort of raising this possibility that these effects might be somewhat stable within individuals. But of course, the big question is not whether or not these effects are stable within individuals over time, but it's whether or not this effect really replicates in an independent sample. So I sort of glossed over the fact that all of the analyses I've been showing you up till now were actually run using a leave one out cross-validation scheme, which is a method that we know can overfit the data. So we also really wanted to have an external validation sample. So thanks to our collaborators, we managed to cobble together a heterogeneous replication sample. I'm not going to go into all the details of this sample today, but it was 45 individuals, about a third of whom were taking methadone for opiate use, but we're now seeking treatment for cocaine, as in our original sample. But 2/3 of them weren't on methadone, and they were only cocaine dependent. And they were also scanned prior to a totally separate treatment trial involving a different medication. So again, a very heterogeneous replication sample. And for this sample, we again just create summary scores by summing edge weights, the nodes identified in our original analysis, and entering them into correlational analysis with our biological measure of abstinence, % cocaine-free urines. And when we do this, in fact, we do find the same relationship between network strength and within treatment abstinence in this heterogeneous independent sample. So just to summarize that first data set, these data are really demonstrating the ability of what at the time was a pretty new technique to predict treatment outcomes. In this case, we're successfully predicting abstinence from cocaine during 12-week treatment. I didn't show you the data today, but we've recently replicated this in a second external sample. So we're pretty confident that these effects are real. I also didn't have time to show the data today, but this is just to point out that the networks that we identified were really robust, and they were relatively unchanged when we did follow-up analyses controlling for other baseline clinical variables. So for example, if we control for methadone dose or days of past month cocaine use. Finally, post-treatment connectivity within these networks also predicted abstinence during follow-up, and our networks were not changed from pre to post-treatment, raising the possibility that this relationship may be somewhat stable over time, which is a point that I'm going to return to at the end of the talk. But before that, I sort of want to return to this issue of brain state. So in the second part of the talk, my lab has really begun looking into whether brain states are in fact drug specific. And this was work that was recently published in Molecular Psychiatry and was led by Dr. Sarah Lichtenstein when she was doing her postdoc in my lab. So the clinical rationale for looking at brain state really came from some ecological momentary assessment data from a few years ago. So this was an electronic diary study of methadone-treated individuals with cocaine and opioid dependence, so very similar to our polysubstance using neuroimaging sample. And in this study, what they did was they asked participants to track changes in mood and craving and to document their substance use within an EMA framework. And what they found was a differential association between positive versus negative mood states and future substance use, such that cocaine was most robustly associated with having been exposed to the drug or being in a positive mood state, suggesting potential links with impulsivity and reward. But heroin craving was actually associated with increases in negative affect, suggesting possible links with emotion regulation or inhibitory control. And so based on this finding that different mood states predict opiate versus cocaine use in the same individuals, we wanted to know whether different brain states might more closely link to opioid versus cocaine use in our neuroimaging sample. So for this analysis, instead of focusing on cocaine use, we're focusing on opiate use. However, the primary sample is the same. So if you recall from the beginning of the talk, when I introduced the sample, I mentioned that despite seeking treatment for cocaine use, all of our participants had co-occurring opiate use disorder and were actually taking methadone for opiate use disorder. And in fact, there was actually a fair amount of residual opiate use during treatment in this sample. So for this analysis, again, we're doing pretty much exactly the same thing. We're calculating functional connectivity matrices. However, this time we're doing this using the data acquired during a different brain state. So we're doing it using the data acquired during cognitive control task performance. And we're entering those functional connectivity matrices into our brain behavior model, along with our biological measure of abstinence, which here is the percent of non-methadone opiate-free urine submitted during the 3-month treatment. So here on the y-axis, we have the individual participant abstinence values as predicted by our brain behavior model. And on the x-axis, we have the actual abstinence values for each participant. So again, as a reminder, typically when we're using correlation, we're trying to explain the variance between two variables, but here we're just using it to characterize predictive accuracy with a correspondence between actual and predicted values. And so as you can see, our model has relatively good predictive accuracy with a Spearman's error of 0.34, which means that about 12% of the variance in within treatment opiate use is accounted for by connectivity within our network. And for context, that 12% amount of variance is comparable to the amount of variance explained by similar models that I showed you at the beginning of the talk that we're seeking to predict trait-like behaviors such as IQ. However, here we're actually predicting a future clinically complex behavior. As with the cocaine network, our opiate network is complex and it includes connections between multiple adjacent and non-adjacent brain regions. And in fact, our opiate network is somewhat larger than the cocaine network. So together, the positive and negative networks include just under 1,000 edges. So it's almost twice the size of the cocaine network. However, that's still less than 3% of all possible connections. Again, indicating that even though there's a lot of visual complexity, these are actually quite specific connections. So to begin to understand the anatomy of these connections, we can again summarize them via their overlap with canonical networks. And when we do this, we can see that at least relative to the cocaine network, the opiate network is somewhat more sparse in that it doesn't include any within network medial frontal or default mode connections, for example. When we directly compare the positive versus the negative networks, we also find that the positive network includes relatively more within network connections for the motor sensory networks, whereas the negative network is characterized by more connections between motor sensory and frontal parietal and default mode and medial frontal networks. I do have to say that we were somewhat surprised by the large number of motor sensory connections here. And so we've also taken a virtual lesion approach with this data, in which we're basically just knocking out all of the connections that overlap with different canonical networks. And when we do this, we find that despite the seeming centrality of this motor sensory component, the overall opioid network still predicts abstinence if we remove all of the motor sensory connections, or in fact, if we remove all of the connections from any single other canonical network. For example, if we remove all the default mode connections. So really what that's indicating is that no single canonical network alone is required to support opiate abstinence, but rather it's this sort of distributed process across multiple complex networks. So to facilitate understanding, we've again proposed a simple theoretical network model that summarizes key aspects of this network. So this figure is just emphasizing that abstinence was associated with increased within network motor sensory connectivity and increased between network motor sensory to salience network connectivity, as well as increased between network default mode to frontal parietal connectivity, as shown by the red lines in the figure. And then the model is also emphasizing that abstinence was further associated with decreased connectivity between the motor sensory network and medial frontal default mode and frontal parietal networks, as indicated by the blue lines. And again, our hope really is that by summarizing the data in this somewhat simplistic manner, we can encourage others to test these theories in their own data sets to further guide neurobiological discovery. Because we're interested in mechanism, we were, of course, very interested in the anatomical overlap between the cocaine and the opioid networks. Interestingly, when we compared the connections across the networks, we actually saw very little overlap, so less than 1% of the edges were shared. So here on the left, you can see edges that were common to both cocaine and opiate networks, with red lines indicating shared positive edges and blue lines indicating shared negative edges. And as you can see, there really aren't many shared edges at all. In addition, we also identified several edges that have opposite associations with opiate versus cocaine use. So for example, these are edges for which increased connectivity predicts cocaine abstinence on the one hand, but for which decreased connectivity predicts opiate abstinence, or vice versa. And actually, there were slightly more opposing edges than there were consistent edges. And as I said, very little overlap overall. So really, these data are indicating that the neural substrates of cocaine and opiate use may be largely dissociable, even in the same individuals. So we've also been looking into the specificity of these networks for predicting specific drugs across different brain states. So the data I just showed you indicate that the cocaine network and the opiate network have pretty limited anatomical overlap. However, as you may recall, one network was derived from reward task data, and another was derived from cognitive task data. And I haven't yet shown you whether opiate networks are also related to cocaine use or vice versa. And I also haven't yet presented data to determine whether the relationship between network connectivity and substance use is in fact task dependent, or in other words, whether or not our connectivity findings are holding across brain states. So to answer this question, first, we looked at the impact of brain state on network identification. So we repeat our connectome-based model of opiate abstinence, this time using reward instead of cognitive task data. And when we do this, we found that we were only able to predict opiate abstinence using the cognitive task data. Similarly, when we repeat our connectome-based model of cocaine abstinence, this time using cognitive instead of reward task data, we find that we're only able to identify the cocaine network using the reward task data. And so together, this is really demonstrating that the identification of both opiate and cocaine abstinence networks is brain state specific. So having established that, next, we wanted to test whether once identified, the relationships between networks and specific substances might hold or generalize across brain states. So here on the left, you have the association between within treatment opiate abstinence and connectivity within the cocaine network. And you can see that there's no significant association. And then on the right, we have the relationship between cocaine network connectivity and abstinence across different brain states. So for these analyses, we're taking the cocaine network that we identified using reward task data, and we're using it as a mask to extract connectivity during cognitive task performance. And what we find is that we again see a modest linear relationship between network connectivity and within treatment cocaine abstinence. And then we also see on the right that we see a similar pattern when we look at connectivity during resting state. So together, these data are really indicating that while the cocaine network does not generalize to predict while the cocaine network does not generalize to predict opiate use, the relationship between cocaine network connectivity and cocaine use does in fact generalize across brain states. We see a really similar pattern when we look at the opioid network. So here on the left, you have the association between within treatment cocaine abstinence and connectivity within the opiate network, this time during cognitive task performance, and you can see that there's no significant association. Whereas when we take the opioid network that we identified using cognitive task data and use it as a mask to extract connectivity during reward task performance or during resting state, we again see a positive association between network connectivity and within treatment opioid abstinence. Again, indicating that while the opioid network does not generalize to predict cocaine use, the relationship between the opioid network and opioid abstinence does generalize across brain states. I don't want to overly belabor this point, but this is just to show that we've run all possible combinations of this. So for example, we've looked at whether opiate network connectivity during resting state relates to cocaine use, and in all cases, the specificity of these effects remain. So in other words, we're seeing this double dissociation such that the cocaine network is consistently unrelated to opiate use and vice versa. As with the cocaine data, we've also looked into the relationship between connectivity within the opiate network as measured post-treatment and subsequent opiate use during 6-month follow-up. And when we do this, we again find a similar relationship between post-treatment connectivity and abstinence following treatment, potentially indicating some consistency of this relationship over time. And also when we compare connectivity within the opioid network from pre to post-treatment, we also don't see any significant changes, further suggesting stability of these effects. Which is interesting, and to me touches on a really important point, which is that networks contributing to treatment response may actually be distinct from those that change with treatment or that are directly implicated in disease pathology. It's also possible that brain regions which predict treatment outcomes may just be different from those that change with treatment. So at first that can seem counterintuitive, but when you think about it in a clinical context, we know that factors that predict treatment response, so things like motivation to change, can be distinct from things that change with treatment, such as the acquisition of new skills. Therefore, the same may be true of neural networks. Further, it's also possible that changes within the abstinence network may just take time to emerge and thus may only be detectable months after treatment. And that theory is actually really consistent with data demonstrating that abstinence rates continue to improve following some behavioral treatments for cocaine use disorder. So again, it stands to reason that neural change might also take time to emerge. And so we've begun trying to explore that first possibility, or the possibility that our networks are not only predictive of future substance use, but that they also just may be altered relative to healthy control individuals and therefore perhaps linked to addiction pathophysiology more generally. So the theoretical basis for looking into this, for looking into comparisons with healthy controls, comes from a nice paper from Hugh Garavan and colleagues from a couple of years ago, in which they propose a couple different scenarios for what prolonged abstinence might look like at the brain level. So here at the top, they propose that one scenario would be that recovery from addiction could involve some restoration of premorbid brain function. In the middle, they propose that an alternative hypothesis could be that prolonged recovery may in fact require some sort of hyperfunctionality of brain regions involved in abstinence maintenance to above the levels observed in healthy controls. Or finally, at the bottom, they propose that a third scenario is that individuals in recovery from addiction may just continue to exhibit decreased function, thereby conferring continued vulnerability for relapse. So just to unpack that a little bit more, based on that theoretical model, if we were to compare our abstinence networks to healthy controls, and we found that abstinent individuals had similar levels of network strength relative to healthy controls, we might conclude that what we're seeing is a return to premorbid functioning. Alternatively, if we were to find that individuals who achieve abstinence have increased network strength relative to controls, we might conclude that what we're seeing is an elevation of brain function or a hyper recovery. And finally, if we were to compare abstinent individuals to healthy controls and found that they had decreased network strengths, we might interpret that as indicating a continued vulnerability for relapse. So to test those theories, we've computed connectomes from identical task data for 38 age and sex matched non-substance using control participants. And we just compared this to our polysubstance using sample. So here at the moment, I'm just showing you a binarization of the data I presented earlier. On the left, we have cocaine network strength for individuals who achieved some abstinence from cocaine during treatment. And on the right, we have network strength for individuals who didn't achieve any. So this is circular to what I showed you before. But what's interesting is when we add in our healthy controls to see where they fall. And what we find is that non-substance using individuals are actually intermediary between responders and non-responders, such that our treatment responders actually have increased network strength relative to controls, and our non-responders have decreased network strength relative to controls. We again see a really similar pattern when we look at the opioid network. So we find that our control group is again intermediary between responders and non-responders, with responders having increased network strength relative to controls. Although the differences here between controls and non-responders is not significant. So for both the cocaine and the opioid networks, we're seeing this pattern that treatment responders have greater network strengths than control participants, which would really be consistent with this notion of an elevation of function relative to controls, or sort of hyperfunctionality. So most recently, we've actually been applying the same approach to try to identify predictors of cannabis use outcomes, which I think is, a really important issue within the context of the changing legislation in this country. So for these analyses, we're just combining aggregate pre-treatment neuroimaging data from 2 separate RCTs. And by combining data from these 2 samples, we were able to come up with a neuroimaging sample of 58 individuals with primary cannabis use disorder, all of whom were scanned prior to treatment. And for these analyses, we again just have connectivity matrices that we generate from both cognitive control and reward task data. And we enter these into our predictive model along with our dimensional biological measure of abstinence. And surprisingly, unlike with the cocaine and the opioid networks, what we found was that we're actually able to generate accurate predictive models of cannabis use abstinence using both data types, really suggesting that both cognitive and reward-related brain states are relevant for understanding cannabis abstinence. So here on the left, we have the correspondence between predicted and actual abstinence values for the cognitive task shown in gray, and also for the reward task shown in white. And what we can see is that for both types of data, the Spearman's rho is hovering around just under 0.4, indicating that about 16% of the variance in within treatment abstinence can actually be accounted for by connectivity within these networks. As with the cocaine and opiate networks, we also then tested the generalizability of this effect to an independent sample of individuals with primary cocaine use disorder, the results of which are shown on your right. And so this is just showing you that we find that the cannabis network does not generalize to also predict abstinence from cocaine, really further indicating substance specificity of different abstinence networks. We're really just only starting to dig down into the anatomy of these networks. Sarah Lichtenstein, who's the postdoc I mentioned earlier, who's now an assistant professor in my lab, is taking the lead on these data, and I'm writing this up. So I don't have too much to show you just now, but I want to highlight a couple things. So far, the anatomies of the networks identified during our reward and cognitive tasks do have some similarities. So these chord plots are summarizing positive network connections for the networks identified during cognitive task performance on your left and reward task performance on your right. And these chord plots are really similar to the other circle plots I presented previously. But here, instead of summarizing macro scale regional connectivity, they're just summarizing connectivity between canonical networks. And so we can see that for both tasks, the cannabis network is characterized by high degrees of connections between frontal parietal and motor sensory networks, as indicated by these sort of blue to pink arcs. So those look really similar, but we also do see some differences. So for example, we can see some differences in patterns of connectivity related to the salience network and also to visual networks. And those differences are actually even more striking when we compare the negative connections. So when we compare the connections for which decreased connectivity is a positive predictor of cannabis abstinence. Here, what we're seeing are clear differences in patterns and patterns of connectivity between this, for example, salience and motor sensory networks. We can also see that during cognitive task performance, the model is identifying a number of connections between default mode and medial frontal networks, as indicated by this sort of green arc, which we're just not seeing in the reward task model at all. So as I said, these are new data, and we're really still working on drilling down into the anatomy of these networks. But I think that, these data are really, again, highlighting this idea that brain state may be a significant factor for generation of optimal models. So even if we're trying to predict the same behavior in the same individuals, the connections that we can identify seem to be partially dependent on the brain state the participant was in during data acquisition. So that's all of the data that I'll present today, which I think demonstrates both the ability of our approach to generate specific externally valid predictions and also of the approach to provide neurobiological insight. So hopefully what I've demonstrated today is that networks identified using this approach are in fact clinically relevant. That is, they're able to predict treatment outcomes. These networks are also externally valid in that they're able to generalize to predict specific behaviors in novel samples and individuals. In addition, despite their complexity, these networks are in fact biologically meaningful in that they're composed of specific connections observing specific behaviors. And finally, these networks are really robust to common sources of variance. So they predict even after we control for things like baseline severity or treatment assignment or even medication status. So if you're interested in applying predictive modeling approaches to your own data, I recommend checking out the August issue of Biological Psychiatry, Sienna and I, from last year, which was dedicated to data-driven approaches. This included our review paper that covers recommendations for best practices in cross-validated biomarker research. And that's within the specific context of addictions, but I think that really most of the issues we cover apply to clinical predictive modeling more generally. And that's probably why I figure summarizing the workflow for this type of approach made the cover. In the interest of time, I'm not going to go through all of the recommendations that we outlined in the review, but I do want to highlight a few key points, one of which is really careful consideration of the window of assessment when you're doing this kind of work. So very often scanning prior to any clinical intervention is often considered sort of desirable and the gold standard. But really, it's important to remember that scanning during this time can introduce unnecessary confounds, such as effects of acute intoxication or withdrawal in the case of addiction, or effects of mood or psychosis state in the case of other disorders. In addition, scanning prior to clinical intervention isn't always feasible unless treatment initiation is delayed, and that can, of course, place unnecessary burden on the patient. I also want to emphasize the importance of employing multiple performance metrics for quantifying model accuracy, even though I didn't get a chance to go into that today. And also the importance of setting realistic expectations for effect size estimates for findings from cross-validated models. And then finally, I really just want to emphasize the importance of conducting post-hoc testing to provide elucidation of mechanism. Because without this, again, findings even from models with high accuracy are going to do very little to advance our understanding of the underlying neurobiology, which is really what is essential if we want to inform novel treatment development. So within that context of post-hoc testing, it's really important to consider model findings across multiple levels of interpretation, with the most basic level being the connections themselves, and the most abstract level being the overarching neurobiological model or the theoretical model that can then be used to guide treatment and as a basis for further testing. So within the context of, for example, a simple region of interest-based approach, one method for determining significance of different features is to rerun the model, excluding specific features. This is that sort of virtual lesion approach that I mentioned with the opioid network. And we do this to determine which features are necessary for optimal model performance. So for example, this would let us answer questions such as, does amygdala connectivity contribute to overall model performance? Similarly, you can rerun the model only including selected features, and that will enable determination of the relative weight of specific features. So for example, what is the predictive ability of amygdala connectivity by itself? So these are really simple steps, but people often skip over them. So this is just like my PSA reminder that elucidation of mechanism should also be a goal of prediction. If you want to learn more, I'll refer you to the review as well as to a 10 simple rules for predictive modeling that we published in Neuroimaging a couple of years ago. I'll end there, but I want to thank all of my collaborators, particularly the primary co-authors on the data I presented today, which is shown here in bold. I also want to thank everyone in my lab and the organizers of this session for inviting me to talk, and everyone here for listening.
Thank you so much, Dr. Yipper. Fantastic talk. We're going to open up. We have about 10 more minutes left for today's session. So we'll start with some questions that people have put in the chat already. And then if you have any questions, just make sure to put those in the Q&A box so that I can read them out. So the first question, oops. Okay, the first question is by Melanie Koss, and sorry if I pronounce anyone's names incorrectly throughout this session. And they ask, are the individuals from the 2019 study still being checked in with? Are there individuals that were able to remain abstinent in the 12-week period, but perhaps not further out? basically wondering how far in time this model can account for.
That is a great question. And unfortunately, we are not following those individuals, but we are involved in other ongoing data collection initiatives, where what we do now is we partner really closely with the methadone and buprenorphine clinics that we recruit from. And so, in the context of, longitudinal research, it's really hard to follow someone, for a year or 2 years, in the context of a research setting. But the good news is that, well, I don't know if it's good news, but people stay on these medications for a long time. This is, it's not something they take for a short amount of time. And so by partnering with the clinics, our hope is that we can get much longer term data. So we will know who's still in treatment, 2 years later, and also get medication adherence data on that, because it has to be documented, when they're taking their methadone and everything. But yeah, great question.
All right. And Jocelyn Rickard asks, thank you for your talk, Sarah. Were you surprised by the lack of shared edges between the cocaine and the opioid networks?
I thought there would be more overlap, but then at the same time, the more we sort of started, again, our hypothesis was coming from this cool EMA study from a number of years ago by Epstein, where they found this dissociation between mood states that were predicting decisions to take cocaine versus opiate craving. They couldn't actually predict decisions to take opiates, but they could predict craving, that were really different even in the same individuals. So it's sort of, in that sense, it's kind of intuitive. There's also a lot of, emerging research on differences in, just social aspects of polysubstance use. So cocaine is more often taken outside the home and heroin is more often taken inside the home. So yeah, I think they're just potentially kind of different behaviors. But yeah, it would be interesting to look at it in a more sort of recreational context. Like if you have, college students or, that both recreationally use alcohol and recreationally use cannabis, if it's in a state where it's legal, are similar mood states and also similar brain states related to those decisions. How do you choose between having a beer and, eating an edible, I guess.
Yeah, it makes a lot of sense. P.S. Ragier says, hi, Sarah, great talk.
Hey, Paul.
Paul. The first question they asked is, how feasible are these analyses for relative novices to this type of method? And then I'll ask the second question after.
Great question. If you have connectivity matrices, then CPM itself is actually surprisingly straightforward, and all the scripts are, as I said, publicly available. But I'm happy to chat with people and answer questions and all of that. If you don't have matrices yet, and you've never done some sort of functional connectivity analysis, then it's kind of just like anything in neuroimaging, you just sort of have to learn it. The way that we make our matrices, we work with people here at Yale that use the bioimage suite, and they have their preferred network parcellation, but the code you can do with any sort of network parcellation. And then the tools that the bioimage suite creators have created to help with the visualization. So those pretty circle plots and things like that, you can actually, there's a website that lets you upload your matrices and it creates those visualizations for you. And if you're using a different parcellation scheme, so let's say you want to use, the Powers Atlas or whatever, you can also upload that there. So it doesn't even have to be in the same space as the original parent papers. There's a Nature protocol paper, or Nature Neuroscience protocol paper, with Shen as the first author, from I think 2015 or 2016. And then the very original CPM paper itself was Finn as the first author in Nature Neuroscience, around that same time.
And then his second question was, the tasks were important for identifying outcome relevant networks for opioid and cocaine participants. Previous studies typically use resting state data for large scale brain network data. Did you find that resting data was able to identify the opioid and or cocaine specific networks?
Yeah, also a good question. So we didn't have as much resting state data. I sort of glossed over that. We, the data were actually collected before my time. So I was analyzing a lot of this for part of my K01. But the study didn't start collecting resting state until about halfway through. So we only have resting state data for about 30 of the original 54 individuals. And that's just kind of a small amount to try to build these models. So we haven't really done it. Oh, cool. Someone just put the protocol paper in the chat. Oh, that's helpful. But yeah, I mean, again, there's data from some of my colleagues here that really does suggest that when you're running these kind of models, very often task-based data is sort of, for whatever reason, better at generating predictions. And we don't really know why. I mean, we think we have this analogy that it might be sort of like akin to sort of like a cardiac stress test in medicine. So this notion that, if you like measure someone's heart, I'm not a physician, but if you are checking someone's pulse or whatever, just when they're at rest, they may seem fine, but if you stress them out by putting them on a treadmill, you're able to get a more sort of finer assessment of their cardiac function. And so hopefully our tasks aren't particularly stressful, but they are sort of brain state inducing, right? And actually the anatomy part of it is really interesting. So I didn't have time to go into this, but again, the networks that we identify are super intuitive within the context of the task, even though we're not modeling the task. So like that really high degree motor sensory component of the opiate network, so the cognitive task that we were using was actually a Stroop task. And if you put the word Stroop into neurosynth, what comes back as, one of the most activated regions during the Stroop actually overlaps with that high degree motor sensory node, which is just really interesting.
Awesome. So Lee Yan McCurdy asks, this is such an interesting approach. I was wondering whether there were any behavioral psychosocial variables which, when combined with CPN, improved predictability.
Yes, also a good question. So for the cocaine paper, the reviewers asked us to basically do a similar machine learning approach of the behavioral data. And when we did that, we weren't able to just predict using the behavioral data. I do think that absolutely adding in other variables can help. So part of the problem is that part of the reason we haven't gone too far with that is that you kind of need your external replication sample to test that, right? Because you're identifying the network in one group. And then really you want to take that network and then test it in the external sample along with the other behavioral variables of interest. And we did that in, again, the cocaine data where we had an external replication sample, and just really basically when we do a binary prediction, yes, no, any abstinence during treatment, we find that if we add that, we find that our findings replicate, but we also find that if we then add in things like baseline severity, the model continues to improve. So the more data you have, the more accurate it's going to be.
Awesome. And there's one more question. I know that we have a stop time at 1, but I'll just ask it and we can see if we can make it. Is there any prospect of future research randomizing participants to different types of treatments for substance use disorders to determine if different networks or subnetworks predict continued abstinence depending on the treatment approach?
Yeah, that would be great. I mean, yes, please tell NIDO to fund me to do that. One thing that I think would actually be really cool is to do to stratify randomization groups based on network strength. So going in, that in both arms of say a randomized treatment trial, that the placebo and the active drug group have sort of similar network strengths on balance so that you're sort of controlling for that individual difference factor. But yeah, I think there's a lot of stuff that could be done. And yes, that is one of the many long-term goals.
Awesome. All right, well, we got through all the questions. There's now going to be a trainee Q&A with Dr. Yip that Victoria might like to introduce.
Sure, yeah. So I just posted the link in the chat, but we are going to move over into a new Zoom room, and we invite you to come with further questions that maybe we didn't get to during this session, or Dr. Yip has also said that she's willing to answer questions, science and otherwise, sort of an Ask Me Anything style. So you can meet us over there. We'll be hopping over there now. Thank you so much, Sarah, for your talk. It was really great.
Thanks, everyone.
Thank you.